Welcome everybody after a long summer holiday. The new academic year has started, and as we agreed, I keep supporting your preparations with videos. This is the opening slide, and on the right part you can see some important info about the exam paper itself. Obviously the agile papers, the high-level papers, are different from the standard level ones in some aspects. So first and foremost, these high-level papers have two parts. The first one is compulsory for everyone, and you can get 80 points for this part. And the second part contains two optional tasks. You have to choose one, and you have to complete one for 20 points. It's important to keep in mind that you can't get more points if you work out both of the optional tasks. In case of the multiple choice questions, indicate your answer clearly in the empty boxes as it is being demonstrated in here. If you make a mistake, cross out the wrong answer and indicate the good one next to that, so your corrections should be clear. As for the open-ended questions, write your answers always onto the dotted lines and pay attention to grammar, please. After every single exercise, you can see these gray boxes. These are for the teachers. You don't have to write anything here. And what is important, black or blue ink should be used. And please don't forget about our books. So these 3 books help you to revise the material for the final exam, all of them based on the requirements. So these are extremely useful pieces of information for you. And the first question is entitled the pelvis. In the picture, the bones of the human pelvis can be seen. Name the bones indicated by capital letters. This little image will help us to answer the questions. Let's start with A, which is showing us the bone structure which is located at the base of the lumbar vertebrae, and it is called sacrum. B is the uppermost and largest bone of the hip, and the name of it is ilium. C is indicating a bone which is located in front of the pelvic girdle, and it is called pubis. Then D is going to be the ischium, which is the lower back part of the hip bone, and E is the last region of the vertebral column. This has a little triangular shape, and it is called coccyx, which is commonly referred to as tailbone. Name the type of articulation formed between bones B and C. The articulation that can be found in between these bones is a kind of fusion, bone fusion, a fixed articulation, fixed joint. Exercise 3. The nerve fibers of which division of the automatic nervous system leave the segment of the spinal cord protected by bone A? Where are the cell bodies of these postganglionic neurons found? Okay, so we have to answer 2 questions in here, and then we learn the autonomic nervous system, we learn the structural and functional differences in between the parasympathetic and sympathetic nervous system. And we know that actually they come from different regions of the center of the spinal cord. The sympathetic nervous system is originating from the middle part of the spinal cord, and the parasympathetic nervous system is from the brainstem or from the bottom of the spinal cord. That's why the good answer here, the first good answer here, is going to be the parasympathetic nerves. And to answer the second question, which is about the location of the ganglia, we know that the sympathetic nervous system ganglia can be found closer to the spinal cord. Why the parasympathetic nervous system ganglia will be closer to the target organs. So the next answer will be the following. The cell body of postganglionic neurons are found in the wall of visceral organs. One of the visceral organs located in the pelvis is the rectum, which communicates with the outside through the anus. Name the other body openings in the pelvic region. In both men and women, we can find an opening which is called the urethra, and only in women there is an entrance which is called the vagina. Due to the wear and tear of severe inflammation of the articulating joint surface socket, marked by F, here you can see it, hip replacement surgery might be necessary. The result of the surgery is shown in the photograph. Name the long bone, part of which was replaced in the surgery. Very easy question. This is the longest bone of the human body, which is called femur. A frequent but treatable abnormality of the pelvis is the dislocated hip. Name the characteristic feature of the pelvis which increases the likelihood of having the hip joint dislocated. We know that the socket is part of the pelvis, which can also be called acetabulum, but the socket is extremely shallow, and therefore the femoral head can be easily dislocated from that, so it should be written down somehow. The socket is shallow, so the head of the femur is easily dislocated. That's it for the first exercise. Let's continue with the second one. Phenylketonuria. Phenylketonuria is an autosomal recessive heritable disease which can be traced back to the lack of an enzyme. If left untreated, it causes severe mental retardation. In Hungary, an average of 1 out of 9,000 children is born with phenylketonuria. The disease is caused by a defect in the enzyme called phenylalanine hydroxylase. The function of the enzyme is to convert the amino acid phenylalanine into tyrosine, another amino acid. Due to the lack of this enzyme, phenylalanine cannot be converted into tyrosine in the liver, and as a result, phenylalanine along with some of its derivatives accumulate in the blood. High concentrations of phenylalanine damage the myelin sheath of nerve cells. The most frequent mutation, which occurs in the gene PAH coding for the enzyme, causes the substitution of one single amino acid for another in the enzyme protein. Instead of tryptophan, arginine is built into the amino acid chain. The damage to the nerve cells can be prevented by an appropriate diet, which contains the minimum required amount of phenylalanine. Screening test for the disorder after birth is of vital importance and has been compulsory in Hungary too for a long time. Based on the information provided in the text and with the help of the Codon dictionary attached, write up the original correct and the mutated, due to one base substitution, base pairs of the triplet involved in the amino acid substitution. So let's look at this Codon dictionary that will help us to figure out the mRNA codons. So what kind of nitrogenous bases will build up tryptophan? We have here tryptophan, so it's very easy to figure out that it is going to be UGG. And from this codon, actually, the DNA active strand and silent strand can be figured out. So the silent strand will be TGG, and then the active strand is ACC. Okay. And as for the mutated allele, we are looking for arginine. And if you look at the Codon dictionary, you can see the well-known fact that more than one codon can code for one amino acid. So here you can see that actually many different, slightly different mRNA triplets can be used in translation in order to have arginine. So we know that only a base substitution mutation took place, which means that one base has been changed. In this case, actually, we have two options. The mRNA can be CGG or AGG. Now, out of these two options, it's very easy to figure out the active strand and silent strand. So the active strand can be G or T, obviously CC, and then the silent strand will be C or A, and then G. I hope it makes sense. So these were the mRNA triplets. Based on the excerpt and your own knowledge, state whether the phenylalanine is an essential or non-essential amino acid. Give your reason for your answer. From our background knowledge, we know that non-essential amino acids are the ones that can be synthesized by our bodies, and then essential amino acids come from our diet, so we need to consume them. Therefore, it is an essential amino acid, and the reasoning is nothing more than the previously mentioned statement, so it is not synthesized in the body, it must be eaten. Fill in the gaps of the following text, which offers a possible explanation for how the substitution of an amino acid could result in a defective enzyme. Okay, so let's fill in the gaps. The substitution of amino acids can alter the structure of a globular enzyme proteins to such an extent that molecule, as defined, cannot fit into the of an enzyme. Very, very easy task. We know globular proteins have tertiary structures, then the substrate can't bind to the active site of the enzyme. As a result, the enzyme cannot convert the phenylalanine into tyrosine. Geza is healthy, but both of his grandmothers, paternal and fraternal, suffer from phenylketonuria. Geza's parents are both of healthy phenotype. Geza's partner, Hannah, is also of healthy phenotype and her parents are healthy too. However, Hannah's brother, Peter, was also diagnosed with phenylketonuria at birth. The couple would like to be informed about the chances of them having a child suffering from phenylketonuria. Let us assume that in the above case, those suffering from phenylketonuria do not show the symptoms of the disease because of their strict phenylalanine diet. As a genetic counselor, support your answer with calculations. Okay, this is a very nice exercise, and if you take my advice, first draw everything that you can read out from the text. So we know that the disease itself is autosomal recessive, that's a big help. And let's start from Geza. So Geza is healthy, and we know that the parents were also healthy, but both of the grandmothers were diseased. I quickly draw it. That's a big help. So their phenotype were the following, small a, small a. It means that the parents were carriers, actually. They were heterozygous for the trait, and we don't know anything about Geza. Let's move on to Hannah. Hannah is also healthy, and her parents are also healthy. So Hannah has parents, obviously, and they were healthy too. But we know a very interesting fact in here. Hannah's brother Peter was also diagnosed with phenylketonuria. So Hannah was one child, and then they had Peter too, and Peter was diseased. If Peter had the disease, it means that the parents must be heterozygous here too, because that's the only possible way how they could donate the recessive alleles to the son. Okay, so the big question is, what do we know about Geza and Anna, and what can we say about the probability, what is the probability of them to have a diseased child? So let's look at their phenotypes. Due to the fact both of their parents were heterozygous, obviously, this would be the constructed Punnett square. But immediately we have to cross out this one from the list, because we know from the text that both Geza, and Hannah are healthy. Okay, so the probability of Geza being heterozygous is actually 2/3, and the probability of Hannah being heterozygous is also 2/3. So we have to write down in a nice way that the probability of Geza and Hannah being heterozygous is 2/3, because their parents are also heterozygous. The next question that must be answered is that what is the probability of their child being ill if they are heterozygous? That is going to be 1/4. Just look at this Punnett square again. And then the likelihood of the couple to have a diseased kid is going to be 1/9, because you just simply multiply the fractions to 2 times 2 times 1, it's 4, 3 times 3 times 4, it's 36, divided by 4, so actually 1/9. Okay? That's the solution. Exercise 3, the domestication of dogs. In the following excerpt, you can read about the domestication of dogs it thought to have taken place. Answer the questions based on the excerpt and your knowledge. The dog originates from the domestication of a subgenus of wolf, which has become extinct by now and is supported by mitochondrial DNA data. The time of the beginning of domestication is surrounded with scientific debate, but it is thought to date back to 10,000 to 100,000 years. However, since there are no surviving documents about the beginnings of domestication, we can only rely on assumptions. An approximately 33,000-year-old dog mandible was excavated in the vicinity of Oberkassel, Germany, which might have been the remains of the earliest domesticated animal known to date. A skull was also found in Siberia. The genetic variation among dog breeds is no less than the variation between the dog and the wolf, 0.2%. The dog mandible mentioned in the text is 33,000 years old. The age of the finding can be determined by radiocarbon C14 dating as well. Which are the characteristics of this method? Write the letters of the correct answers in the boxes. So let's try to remember what we have learned about radioactive carbon dating. So what you need to know is that radioactive nuclei can decay at different rates, and it is depending on their half-life. In case of carbon, it's 5,730 years. So actually, we can use the half-life in order to determine the age of old objects. How? Carbon-14 is a radioactive isotope, and you have to know that carbon can be found in all of the living organisms. Carbon-14 is present in trace amounts, so in very, very small amounts in living organisms, but the ratio of carbon-12 and carbon-14 is informative because it is going to be the same in the object like in the atmosphere at a given time. So when something dies, actually, the carbon-14 over time start to decay, start to go through beta emission, and it turns back to nitrogen-14 slowly. It means that the carbon-14 content becomes less and less, okay? So it is constantly decreasing. So this kind of information can help scientists to determine the age of certain objects. So let's look at the statements. The older the sample, the less carbon there is in it. That's not really the good answer. It is not suitable for examining samples with high carbon content. It measures the activity of the decaying radioactive isotope. That's what we discussed, so C is going to be the first answer. That is correct. It is based on the fact that with the metabolism coming to an end, the proportion of carbon isotopes is not changing. Actually, it is changing, so it is not a good option. The total carbon content of the sample also has to be determined during the measurement. That's true. And it yields important data about the age of the oldest fossils as well. Not rare. Find one indirect and one direct piece of evidence in the text for the evolution of the dog. Indirect piece of evidence, it is mentioned right at the beginning, the mitochondrial DNA data, and the direct piece of evidence is the mandible itself, so the 33,000-year-old dog mandible. What are the characteristic features of the process giving rise to different dog breeds? So let's look at evolution a little bit. Only the phenotypes have changed. Obviously, this cannot be true. Genes must be changed in order to have different dog breeds. It is the result of divergent evolution. I have a feeling this is going to be one of the good answers, considering the fact that divergent evolution is describing the process by which species from a common ancestor become more and more distinct from each other due to selection pressures. Just remember what we have learned about homologous organs. It is the result of convergent evolution. Obviously, it is wrong. Convergent evolution is describing how the distantly related organisms evolve similar traits like the analogous organs, like the wing of a bird and the insect. That was the example that we analyzed a lot. It does not alter the allelic frequency of populations. Again, this is not true. There is selection pressure at work. I have already mentioned it when we were talking about divergent evolution, so E is the other good answer. Perfect. Let's move on to exercise 4. Mitochondria. Mitochondria ensure the energy supply of the cells. Choose from the list of words below to fill in the gaps of the text explaining the figure. You do not need all the words. This little graph is helping us a lot in order to answer the questions or fill in the gaps. So the end product of D enters the matrix of the mitochondrion. We know that glycolysis is running in the cytoplasm, so actually that must be glycolysis. End product of glycolysis is pyruvate, by the way. After going through changes, this molecule enters the citric acid cycle. In the link reaction, pyruvate will be converted into acetyl coenzyme A. So enters the citric acid cycle as a 2-carbon unit. Okay, so glycolysis and 2-carbon unit. Once it has been fully oxidized, it leaves the cycle in the form of carbon dioxide. The high-energy electrons and protons originating from the carbon-hydrogen bonds of the intermediate substances are transferred to the nicotinic adenine dinucleotide, so NAD electron carrier coenzyme. Give the name of the letter which indicates this coenzyme. We know that that's a hydrogen carrier, and this is going to be indicated by letter B. The chemical DNP makes the inner membrane of the mitochondrion permeable for the protons, thereby preventing the buildup of a high concentration of protons concentration gradient in between the inner and outer mitochondrial membrane. After adding DNP to isolated mitochondria, the following changes could be detected. Less ATP was produced, more heat was released, and the oxygen consumption remained the same. So, let's quickly remember the last step of aerobic cell respiration, which is oxidative phosphorylation, and then after that chemiosmosis. But what is important? Basically, the reduced NAD plus another molecule FADH2, but it is not mentioned in here, so the reduced NAD will give electrons to electron carriers. As the electron carriers are moving, they will initiate protons to be pumped to the intermembrane space. That's a very important step in here. We will have many protons accumulated in the intermembrane space. Why? Because we will need them to generate ATP. ATP synthase enzyme can only work with these protons. These protons will move through the ATP synthase and that will generate ATP production. So actually the last step of the biological oxidation will be disturbed by this chemical. So choose the correct interpretations of the results and write their letters in the boxes. Less ATP was produced since the absence of a concentration gradient, the energy source for ATP synthesis was diminished. That is definitely true. So A comes here. The absence of concentration gradient causes the electron transport system to stop, which results in less ATP being produced. It is not affected, so not true. The rate of oxygen consumption remained the same since oxygen was needed in glycolysis taking place in the cytoplasm. No. Since the process of electron transport went on unhindered, the rate of oxygen consumption did not change either. The oxygen is the terminal electron acceptor required in the process. That is true. So D comes next to A. And obviously the last statement is not true, but let's read it. Less ATP was produced because the extra heat release prevented the high energy bonds from being formed between the phosphate groups. No. Okay, so these were the good answers. I hope it was clear. This is not a nice X, okay. Let's move on to the fifth exercise. Counting planktons. Some of the living organisms floating in the water, plankton, carry out photosynthesis. Those are called phytoplanktons, while others belong to consumers, zooplanktons. In the figure, some typical floating organisms are shown. The larvae of a glassworm species is marked A. The species C, D, E, and F, and H, as well as the diatoms from I to N, contain green pigments, while species B, G, contain no green pigments, but they do have pigments suitable for absorbing light. The parachute-like O is an encysted unicellular ciliate. The diatom species from I to N belong to the multitude of unicellular organisms of the plankton. After studying the drawing, give the letter of the species the statement refers to. So, colony of prokaryotes, that is B, that's the only option, that can be the good answer for that. Filamentous prokaryotic organism, if you look at the filamentous structures, I mean the long structures, that's the only structure that can be a filamentous prokaryotic organism, so G. Colony of eukaryotes, that's a gift, so C and H can be clearly seen. And then heterotrophic organism with differentiated tissues, it is written down in the text as well as the larvae of glassworm species marked A, so that's gonna be the answer in here, A. In the highly rigid cell walls of species I to N, an element, which rarely occurs in other organisms, accumulates in large quantities. Name this element. This element is the silica. As we discussed it many times. The quantity of phytoplankton and zooplankton was measured in 1 liter of water from the Lake Balaton in summer and a winter day. The density of organisms is plotted on the left graph, A, while the volume of the plankton on the right, B. The striped bars indicate the phytoplankton in each case, while the dotted bars demonstrate the zooplankton. The exact values are shown in the rectangles on the top of the bars. So here you can see everything that is explained in the text. Members of the zooplankton tend to be larger than those of the phytoplankton they consume. How much larger was the average volume of a membrane of a zooplankton than that of a member of the phytoplankton? Write down your calculations. So let's look at a little bit of math in here. Here, average volume of individuals. You take these two numbers, 100 divided by 120,000, and that's gonna give you the average volume of individuals in the phytoplankton. Very small number in cubic millimeters. And then the average volume of individuals in the zooplankton, you are going to need these two numbers, so 65 divided by 1,000, and that's gonna give us this in cubic millimeters as well. And the question is asking how much larger was the average volume of a member of a zooplankton, so this, than that of a member of a phytoplankton. So you have to do a simple division in here. That's a point, decimal point, 0.83. Both of them are sorry, are given in cubic millimeters. If you do the division, actually you are going to get a number which is 78.3, and to answer the question, 78.3 times as much, or times larger. Okay. According to the measurements, the volume of phytoplankton per liter in winter was smaller than the volume of zooplankton per liter. How can it be possible? Give one explanation. I just wrote here plants produce more biomass than animals because I think that's a very obvious answer for this question, but if you check the answer key, you can see that alternative answers would also be accepted, like in winter plants might have died at higher rate than animals, or members of zooplankton might have found alternative organic food sources, and so on. So please always check the key because there are very good ideas there too. Okay, I hope it was understandable. I think that was enough for the first video, so I split this high-level paper into 2 videos. So the rest of the exercises will be worked out in a second video. Finally, allow me to show you this picture that I took another day with my phone when I was running. Running that day was not easy for me. Maybe I was more tired, or I don't really know. But I know one thing for sure. I looked up the sky and I saw this beautiful sunset, and within a second my feelings has changed a lot, and I felt so many positive things at a time, and I felt extremely lucky to have the opportunity to see this beautiful sunset. So what I wanted to tell you with all of these, when things are getting harder or you need to recharge your batteries, please don't forget to have a nice walk in nature and try to notice the beautiful world that is around us. Being in nature is good for you, never forget, okay? So that's the wisdom from me for today. I'm going to upload the second video soon. Keep up the good work, everything will be fine. Bye bye guys.